But if you're using it chronically, could it be the case that you might cause your hair follicles to essentially self-destruct because you're abusing the ISR system? Maybe. Or perhaps it could reach a point where PP405 actually grows hair, but it might just suddenly self-destruct the hair follicle. It could either permanently destroy the hair follicle, or it might just cause it to just decline over time, like we see with some topical anti-androgens like CBO301 and potentially even RU58841, right? Hey guys, before we continue this video, I would like to mention that we now have liposomal minoxidil sulfate on my website, folligens.com, FOLLIGENZ.com, and if the order queue is available and open, you can order it there. We're running it as a cosmetic. There are other sort of botanicals inside of this particular topical that are pretty helpful when it comes to conditioning the hair. So that's at folligens.com, FOLLIGENZ.com. That's FOLLIGENZ.com. Go check it out and maybe even try it out. See you there. Hello everyone and welcome back to yet another video. Now this video is going to be about PP405 and I am so sorry I'm late to the party. Not really late to the party, but within the past 2 months or so, around the time I broke my computer, or maybe it was Windows 11's crazy ass update that broke my SSD, there was also talks about PP405. From CNN, you had all these news articles essentially talking about Trestless and talking about how people are trying to buy similar molecules online to use on themselves. It's been quite the buzz, and for good reason, right? I made a video, I want to say maybe about a year ago now, and in that video, I was very, very hopeful about PP405, and I had some really good things to say about it. But I think there's also another perspective that we should have here, and this is kind of a bit of, I don't want to say a warning, but some new discourse I want to offer into this discussion. And it involves PP405 and its very nature as an MPC, or mitochondrial pyruvate carrier protein inhibitor, and what that essentially means for the health and safety of the hair follicle. So please sit down and hopefully this video might be able to offer some new perspectives. So before we begin, what is PP405? If you've been living under a rock, PP405 is a novel topical small molecule drug identified by UCLA researchers that works by inhibiting the mitochondrial pyruvate carrier protein, also known as MPC, in hair follicle cells. By blocking pyruvate from entering mitochondria, MPC inhibitors essentially force cells to shunt more pyruvate into lactate production. So we need to understand quickly what pyruvate is. So pyruvate is the primary endpoint of glycolysis. And what is glycolysis, you might ask? Well, glycolysis is the initial anaerobic, meaning you don't need oxygen for this process to occur, but it is the initial anaerobic stage of cellular respiration, and it's a very, very important metabolic pathway that breaks down one glucose molecule into two pyruvate molecules, producing a net gain of ATP, also known as energy for our case, and NADH, which is an electron carrier. Now, this process occurs in the cytoplasm of all cells and leads to fermentation under anaerobic conditions, or it can continue into other sort of energy-producing pathways. So where does pyruvate go after glycolysis? Well, pyruvate goes to the mitochondria to be converted into acetyl-CoA, which enters the citric acid cycle, which is also known as the Krebs cycle, under aerobic respiration, and this is where oxygen is present. And this particular condition requires for more energy production. In the absence of oxygen, so in the case of anaerobic respiration, the pyruvate in this case is fermented into lactate in the cytoplasm to refuel NAD+. But remember, we're talking about PP405 here, and PP405 is part of a class of drugs called MPC inhibitors or mitochondrial pyruvate carrier inhibitors. But what is an MPC? Well, mitochondrial pyruvate carrier protein is exactly what the name entails. It is a type of protein that is responsible for binding to pyruvate and transporting it into the mitochondria. Now, if you're inhibiting this process, that means that there's going to be an excess production of pyruvate building in the cytoplasm, right? Because in more typical cases, that pyruvate would be sent to the mitochondria, and over there it would be turned into acetyl-CoA. But not in this particular case, and that is what makes PP405 quite interesting, because researchers, particularly from UCLA, and also you have from other universities as well, found out that by blocking mitochondrial pyruvate carrier protein, this, for whatever reason, seems to serve as a sufficient stress response that somehow activates dormant hair follicle stem cells to start growing hair again. There were various studies, some on mice, and also some ex vivo studies done on human hair follicles that showed during times of anagen growth phase, they noticed an increase of pyruvate, but they also noticed an increase of lactate dehydrogenase. So this sort of pointed to one thing. Is it the pyruvate that's causing the hair growth, or is it this particular enzyme, lactate dehydrogenase? Well, actually, it is both, and it leads to a conclusion of the pyruvate eventually turning into excess lactate, which it is that excess lactate that is reawakening dormant hair follicle stem cells, essentially turning on that anagen cycle once more. And it's even arguably the case that in severe forms of hair loss or even in end-stage androgenetic alopecia, all it is just a very, very long dormant phase, but the hair follicles and their stem cells are still present, they're just dormant, right? So the body somehow loses its ability to regulate this sort of native lactate switch, and by potentially using this MPC inhibitor PP405, you can essentially achieve this sort of reactivation. Now, PP405 and other mitochondrial pyruvate carrier inhibitors is essentially playing with a system called the integrated stress response, or the ISR system, and it influences another aspect called endoplasmic reticulum, or ER, stress activation. So while shifting a cell's metabolism can potentially awaken stem cells, there's also some risks in triggering cellular stress pathways. And one key pathway to consider is, again, this particular pathway that I named, the ISR, the integrated stress response system. Now, the ISR is used as a sort of backup generator. Now, let's say you have a cell and it's not getting the energy it needs, there's some sort of blockade or stress that occurs, it needs to have some sort of redundancy, a fallback, before the cell implodes and dies, right? And that sort of fallback is the sort of ISR system, where it begins to use alternative forms of energy. In this case, if pyruvate isn't sufficient, it will ferment pyruvate and turn it into lactate, instead of taking pyruvate and putting it into the mitochondria for it to turn into acetyl-CoA. If we look at a study by Pai et al., we can see in ex vivo human hair follicles that the ISR is activated by at least 4 sensory kinases, which are HRI, GCN2, PERK or PERK, and PKR. And these again are sensory kinases, which are enzymes that are helping the cells detect environmental or cellular signal changes. And then from there, they typically help trigger a response. And the sensory kinases that I just named, the HRI, GCN2, PERK, and PRK, or PKR, whatever, these are all uniquely responding to different forms of stress. So heme deficiency, amino acid starvation, and then, particularly for our case, endoplasmic reticulum stress. And we'll talk about this more later. But when activated, these enzymes are going to trigger the way certain genes are activated, which changes the way the cell behaves. And in this particular case, in this particular study by Pai et al., The use of a particular mitochondrial pyruvate carrier inhibitor known as UK5099 resulted in the production of a particular protein known as ATF4, a stress-responsive transcription factor protein. Here, ATF4 then drives expression of genes that may help the cell adapt to stress, which could be anything like restoring amino acid levels or protein folding capacity. However, if the stress is severe or prolonged and this sort of adaptation fails, ATF4 can essentially trigger genes that are responsible for apoptosis. And essentially, apoptosis is like seppuku for the cell because the cell is literally going to kill itself. Now, ER stress, which is the accumulation of misfolded protein in the endoplasmic reticulum, is one classic trigger of the integrated stress response system. ER stress is harmful for hair follicles because the cells, especially during the active growth or anagen phase, rely heavily on the proper folding and processing of proteins. Now, protein folding is the process by which newly made proteins assume their correct three-dimensional shape, which is essential for their function. This folding happens in the endoplasmic reticulum, a specialized structure in the cell that ensures proteins are correctly assembled and delivered to where they are needed. Now, when the ER, the endoplasmic reticulum, becomes overwhelmed due to, again, that sort of stress that we named before, the ISR, right, the abuse of the ISR system, you can have all sorts of issues, right? You may have inflammation triggers, excessive oxidative stress, or maybe even a high metabolic demand, because you're always going to be in that state so long as you use that sort of topical drug, right? That state of the ISR, that lactic fermentation, or the fermentation of pyruvate into lactate. And here, proteins may have a higher chance of misfolding or accumulating in an unfolded state. This triggers a cellular defense system called the unfolded protein response, or UPR, which attempts to restore balance by halting new protein production and increasing the capacity for folding and degrading faulty proteins. But again, if the stress becomes too prolonged or too severe, UPR can activate pathways that really just kill the follicle, or the cell rather, outright. So this is something you want to be very careful with to not abuse, and I don't see it being brought up that often when we talk about PP405, right? And we have to acknowledge this as a sort of possibility that, yes, PP405, or even drugs like it, with chronic use, we don't know what the protocol is going to be like in the future, right? Maybe this is something you can use like stamoxidine, where it's like 3 months on or 3 months off, but we don't know how it's going to be used, right? If it is the case that you have to use it chronically, could a sort of prolonged stress, a prolonged activation of the ISR, potentially kill the hair follicle? This is an interesting question, and I think we can get a hint of what's going on by looking at the Pai et al. Study. So in 2024, Derek Pai et al. Released a study titled, quote, Activation of Integrated Stress Response in Human Hair Follicles, unquote. Like I mentioned earlier, this is the ex vivo human hair follicle study where they used a mitochondrial pyruvate carrier protein known as UK5099. Now, in previous hair loss studies that involved these sort of chemical compounds, these MPC inhibitors, they noticed the same thing in human hair follicles, right? MPC inhibition caused a metabolic stress responsive proliferative arrest in follicles, meaning that the normally fast dividing hair matrix, or the hair follicle cells and bulge cells, essentially hit the brakes on cell division. Through transcriptomic analysis, the authors found that UK5099 triggered a gene signature indicative of ISR activation, and it's particularly the transcription of ATF4 and its downstream targets that were upregulated in these follicles. And my editor will essentially put up the heat map over here, right, where you can see the vehicle follicle, the follicle that got essentially placebo, versus UK5099. And the heat map is showing the genes that are activated the most, right, in these particular samples. And you can see the ones that are green are downstream of ATF4 activation. So that means ATF4 is activating certain genes that are responsible for triggering and producing the ISR environment. Now, this is compelling evidence that the growth arrest was indeed caused by ISR signals. So, in other words, this study showed signs that when we abuse the ISR system, you can essentially kill the follicle. In fact, relatively high doses of UK59 were exposed to human hair follicles for 2 days, which likely represents a worst case sort of stress, right? You have an abundantly high amount of this mitochondrial pyruvate carrier inhibitor that you're continuously exposing to a single follicle for literally 48 hours straight. But it points to the fact that under that particular condition, and theoretically maybe if you were to use a different molecule and if you were to use it chronically, daily, right, once a day, you could potentially have a mirroring effect, right? So the authors talked about how the use of UK5099 made the follicle enter into a regression phase in what was supposed to be its anagen phase. And they call this a dystrophic anagen, which is also known as anagen effluvium, but typically we would call this an early catagen phase, right? So we can summarize it like this. Too much metabolic stress could have the opposite of the desired effect. It might just stifle hair growth or even really just kill the follicle. So to me, I'm hearing that there's going to be a phase 3 starting sometime in 2026, which is, it's pretty good news. But I'm wondering how long this phase 3 is going to last, right? Would it be 3 to 5 years alone? Do we need to see long-term data on the safety and efficacy when it comes to PP405? Because, again, if this dose, which I think currently the dose in clinical trial testing for PP405 is like 0.01%, correct me if I'm wrong, but if the dose doesn't have to necessarily be high, right? The dose doesn't need to be high for it to be dangerous. And I'm not saying dangerous in the sense that it's going to kill somebody, but I'm saying dangerous in the sense that it might just destroy the hair follicle. Maybe the dose is all right, but you have to be careful for how long you use it. Maybe, again, you have to cycle it like stamoxidine, 3 months on, 3 months off. Is it the case that we can chronically use this sort of system and abuse the ISR to give us continuous hair growth? I guess my question is, will there be a sort of blowback effect that eventually you can be using PP405 for 3 years and then all of a sudden, poof, it kills your hair follicles, right? That could be the case. We don't know, but the only way we're going to find out is with more testing. And this kind of brings me back to the whole PP405 48-hour check-in leak that we saw mentioned, I want to say maybe 8 months ago. Now, this part is not substantiated by the Pelage team. It's not even marketed as an official sort of picture by the Pelage pharmaceutical team, but I think there's some credibility to it. So I'm just going to talk about it. Many of you guys have probably heard the news on this already, but hey, let's just see where this is going, right? So earlier this year, there was a leak by a supposed PP405 clinical trial participant known as Chad, and this leak was posted to a Discord server for the NeoStem hair loss product. Now, his post eventually got mirrored by the Hair Loss Cure 2020 website admin. It was revealed by this supposed person named Chad that after 2 applications of PP405 gel, he was called back to the clinic 36 to 48 hours later, so the clinical trial team could take another look at his scalp. And he assumed, and maybe a little too hopefully, that they were checking for signs of new hair growth. And hey, maybe they were, but it feels a lot more likely that they were investigating to see if maybe PP405 caused any irritation to the scalp, or maybe if it killed any follicles. Because I want you to remember, right, if we go back to the Pai et al. Study, what did it reveal? Well, after 48 hours of exposure to UK5099, another potent mitochondrial pyruvate carrier inhibitor, right, similar to PP405, After 48 hours, the follicle was showing signs of it dying. It was showing signs of apoptosis and stress, right? So this could be the case that the pelage team were maybe trying to see, hey, is this guy's hair follicles dying? Oh wait, it didn't die? Okay, well that's pretty cool, right? Now, I already made a video on this particular topic here about Chad's PP405 leaks and how members of my Discord server became essentially detectives, where they found out that these before and after quote-unquote pictures were not before and after pictures, but pictures taken on the same day and of different parts of the scalp. Now, we have this cool guy named G. Nathan on the server who actually, took it to SketchUp to align and to see the angle of the follicles. Very dedicated stuff. And I think another user named Z also went on to do some investigations himself, and he found some pretty decent information. So I want to say thank you to those guys for being absolutely killer with this sort of information. But given these findings, I think it's pretty clear that the UCLA team and the Pelage Pharmaceutical team approach PP405 with both optimism and caution. Even Dr. William Laurie himself, one of PP405's co-inventors, admitted they initially feared that they were pushing follicles too hard metabolically and that they might, quote-unquote, kill all of the follicles, a dire outcome for any hair loss treatment, to be honest. He says, quote, "We were really happy to be wrong about that," unquote, Laurie said, reflecting the relief that their early tests did not show, or rather result in any follicular destruction. But at the moment, it doesn't seem PP405 is toxic, at least in short-term use. Their biopsies at Pelage show that there are appearances of new hair germs, which are the primordial structures that indicate dormant hair follicles entering a new growth phase. So it's literally capturing the transition from the telogen to the anagen phase. But nevertheless, we still need to have some sort of long-term data, right? Whether it's 2 years, 3 years, or 5 years, there needs to be some sort of consistent long-term data that says, hey, if you're applying PP405 for X amount of time chronically, whether or not it's even the case you need to apply it chronically, right? You have to keep that into consideration too. But if you're using it chronically, could it be the case that you might cause your hair follicles to essentially self-destruct because you're abusing the ISR system? Maybe. Again, let's go back to the Pai et al. Study, right? After 48 hours of exposure of UK5099, just 2 days, these hair follicles entered into dystrophic anagen, or anagen effluvium, guys. So that's happening because something is literally saying stop growing in a period of time where the follicle should be growing, right? It's stopping the growth of the hair follicle literally in the middle of its anagen phase. And you actually typically see this with some chemotherapy treatments, because chemotherapy treatments typically target fast-growing cells in your body, and aside from cancer, right, cancerous cells, your hair follicle cells are a class of fast-growing cells too. Now, I'm not saying PP405 is causing cancer, I'm just giving you guys an example, because anagen effluvium is associated with chemotherapy-based hair loss. So, what's the takeaway here, right? The takeaway here is that, maybe we should have a sort of measured look at PP405, right? And I'm not debunking any of the hype, or I'm not saying this drug doesn't work, but I think there needs to be a sort of open discussion about how this drug is potentially going to be used. And that's exactly what Pelage is investigating, right? Is it the case that we have to use this mitochondrial pyruvate carrier inhibitor chronically, this potent one? Do we have to use it chronically? And if so, is that sort of chronic use and abuse, quote-unquote abuse, of the ISR system going to end up in harming the follicle, right? Could it be the case that over time the hair follicle will adapt and there might be some sort of hermetic effect where a small amount of stress yields a beneficial adaptation? Maybe that could be the case. Maybe PP405 might make the hair follicle even stronger. Or perhaps it could reach a point where PP405 actually grows hair, but it might just suddenly self-destruct the hair follicle. It could either permanently destroy the hair follicle, or it might just cause it to just decline over time, like we see with some topical anti-androgens like CBO301 and potentially even RU58841, right? These are all sort of fair questions, and hopefully we get some answers to that, but I'm still going to have my excitement for PP405 because I think it holds a real promise. So, with that being said, thank you guys for watching this video, and let me know what you think about PP405 in the comment section below. But before you go, I have a website and a company that I started called Follicles Therapeutics Corporation. You can go to folligenz.com to go check out a new cosmeceutical that's on the market called minoxidil sulfate, or liposomal minoxidil sulfate. By popular demand, we were able to work to try to get this out, and now you can get this as a cosmetic from my website, folligenz.com. That's FOLLIGENZ.com. If you go to the shop section, you can see that we have 10% concentration of liposomal minoxidil sulfate at 60 milliliters and 5% concentration of liposomal minoxidil sulfate at same 60 milliliters, right? So try it out. Let me know how it goes, and be sure to take pictures and join our Folligenz Discord server in the description below. Anyway, thanks for watching this video again, and I hope to see you in the next one. Peace out.